In this video, I'm going to tell you about the COVID-19 causing virus diagnostic tests being used nowadays. The diagnostic test for COVID-19 causing virus can be divided into two major categories. After the isolation of the genetic material of the virus, that is RNA, it can be subjected to two types of tests. Number one, diagnostic test, and the other one is antibody test. In diagnostic tests, we have polymerase chain reaction and antigen assays, and in the antibody test, we have the antibody test. Polymerase chain reaction for COVID-19 causing virus detection. RT-PCR is mainly used. Viral genomic material is ribonucleic acid and the RNA is reverse transcribed to form deoxyribonucleic acid with the help of enzyme reverse transcriptase. Here is a diagrammatic representation of what I just told you. So we have the RNA and we make DNA with the help of reverse transcription, and the enzyme that is used is the reverse transcriptase, and then we can perform the PCR. So the samples used for COVID-19 causing virus detection through PCR can be either nasopharyngeal swabs or the respiratory tract specimens from the throat swab and saliva. SARS coronavirus 2 genes used for COVID-19 disease detection by PCR can be viral envelope forming gene, viral nucleocapsid gene, viral spike protein gene, open reading frame 1, RNA-dependent RNA polymerase. So now the antigen assay for COVID-19 causing virus. So this assay has relatively lower sensitivity than PCR testing. Antigen test determines protein or proteins of the virus, the SARS coronavirus 2, like nucleocapsid protein, spike protein, and several others. Now here is a test that indicated the qualitative detection of the nucleocapsid protein antigen from SARS coronavirus 2 in nasopharyngeal and nasal swab specimens. Now let's talk about the antibody test for COVID-19 causing virus. Antibody test measures the human body immune response. In response to every infection, human body makes antibodies. Human blood sample is used for the detection of antibodies. Antibody tests are qualitative in nature and provide information to the level of yes or no, while PCR tests can also measure how much virus a person is having at the time of taking sample. Methodology used for the detection of antibodies in the human blood sample is known as enzyme-linked immunosorbent assay or ELISA. Now let me tell you the difference between all the diagnostic tests that I just told you about. If you take a look at this graph, the PCR-based test, it can detect small amounts of viral genetic material, so a test can be positive long after a person stops being infectious. Rapid antigen test detect the presence of viral proteins and can return positive result when the person is most infectious. And the antibody test, indicated by the blue lines in the graph, can detect the body's immune response to the virus and are not effective at the earliest phase of infection. I would also like to share with you emerging technologies in SARS coronavirus 2 detection, the causative agent for COVID-19. CRISPR technology-based tests that include Sherlock test and several other tests. The CRISPR recognized viral genetic sequence, cut the sequence with a reporter molecule. Reporter molecule added reveals the presence or absence of virus. If you want to know more about the information that I shared with you in my presentation, here are the few references. Thank you for watching this video created by Biomed Global, an organization committed to enhancing science literacy across the world.